What if we could detect subtle biological changes before symptoms appear, before disease manifests, before aging overtakes health? EnsembleAge, a new generation of epigenetic clocks, may be the most sensitive and translational tool for that job yet. Welcome back to Decoding Longevity, where we explore the most innovative breakthroughs shaping the future of aging science. I'm Raghav Segal, here with my co-host Maxwell Jakis.
Hey everyone! If you have been following our episodes, you know that we have covered analytics, progeny models, reprogramming, tools that try to reset aging. But what about measuring it? Today, we're diving into EnsembleAge, a multi-clock framework that enhances the accuracy of epigenetic age assessments across stress and rejuvenation interventions.
Joining us is Dr. Amin Haghani, first author of this landmark paper and one of the leading scientists in the field of epigenetic biomarkers and cross-species aging clocks. Amin is a scientist at Altos Labs and currently works with Steve Horvath, previously at UCLA, one of the pioneers in the epigenetic clock field. Amin, welcome to Decoding Longevity.
Thank you very much, Raghav and Mitsu. It's really nice to be here.
Thank you. Thank you for your time. It's exciting. So let's begin with a little bit of giving a big overall picture. So there are many clocks out there, pen tissue clocks, scream age, pheno age, intervention clocks. What prompted you to develop EnsembleAge, and what was the main limitation of the current clocks that you're trying to overcome?
So that's a great question. The truth is actually these clocks, each one of them has its own application. So they have different underlying biology. You can think about it like a blood work. Like you measure different things in your blood. Sometimes you might have to run an fMRI for different applications. So they have different applications. But the problem with aging is aging is multidimensional. So not a single biomarker might not be able to really assess aging. So that's why there is a need to something like an ensemble approach, which will increase the accuracy and sensitivity to assess age-related changes.
So one of the things that you did in the paper was sort of benchmarking clocks against almost 211 perturbation experiments, which is really fascinating. Could you walk us through what this metal gauge data set is and why is it so central to sort of EnsembleAge's performance?
Yeah, that's a good question. So the privilege of working with Dr. Horvath is we will do a lot of collaborations. So after we developed this mammalian array, which is this custom-based array that measure epigenetic signatures in the conserved sequences across mammals. So this platform really can be applied to both mouse and human at the same time. So it really opened up opportunity for lots of collaboration with in vivo scientists. So over the years, we collected a lot of curated a lot of mouse DNA methylation data from different experiments. So that was like the whole idea of the metal gauge came to the play. The truth is the original idea of the paper was called metal gauge. That was the title. But then we changed it because it's like the benchmarking data set is the key, but the data release was so complicated that we made it about the really final product, which was the ensemble clock. So otherwise, like that benchmarking data set was also really essential to develop a biomarker.
Yeah. So just expanding a little bit more on that, just for our listeners. What were those perturbation experiments? So do you mean that you did experiments with the mice and then you collected the different organs at the end, or do you mind just expanding a little bit more to give context?
Yeah. In general, the metal gauge database, which is the curation of different mouse DNA methylation data, it's from thousands of mouse experiments. So these are collaborators. Most of them are aging scientists working on different interventions. That could be chemical interventions. It could be like environmental stress. It could be genetic models. It could be partial reprogramming and many other examples. Disease models. So it's like we had multi-organ data from all of these. So then this was a great opportunity because from those thousands of experiments, I identified 200 of them that we had the expectation. So that means it will affect the lifespan, especially in context of aging. It's relevant to human. The interventions showed efficacy either in the animal models or even in human. Like that could be like calorie restriction or rapamycin. So that kind of thing, it gave a really opportunity to define a set of perturbations and interventions that is relevant to humans. And that's why really that 211 experiments that you are seeing in the paper.
Yeah, okay. So I guess this ties really well with my next question. So you mentioned also that the EnsembleAge itself has a dynamic side and a static side or static mode. Could you unpack a little bit more for our listeners? Because I guess when you talk about dynamic, you also talk about rejuvenation, and rejuvenation, the literally meaning of the word that is to, turn back the clock. Is that like what you see, or is that based on the clock, or how do you define rejuvenation in this context itself?
This metal gauge, so we have a training set of these mice, multi-organ from different ages, comes from the development until like really almost some of them gets to even 3 years old. So we had all this sample. So it was an opportunity to develop the traditional epigenetic clocks. That's what we did though, is like we trained maybe millions of them by limiting to a specific biology, a specific genetic region. So that means it could be like a specific signaling pathways like inflammation, oxidative stress, or actually some of them were only randomly selected. So you can see when you cover different parts of the genome for your clock, each one of them will be relevant to a specific experiments and biology. And when you look at them across these interventions, you see some of them only respond, for example, to exercise. Some of them is like highly sensitive to dietary intervention, some of them reprogramming, and so forth. So when you see this, okay, so then you benchmark them. I benchmarked them. I selected around 40 of these clocks. I called them dynamic. The reason is like, imagine these are a different set of biomarkers. Imagine you have a fMRI, some blood work, some cytokine, like these different biomarkers, right? And you will apply them to different diseases. Now the question is like, can you have one single biomarker that is a surrogate of all of them? So that's what happened. That's when the static came to the play. So the static version, when it changed, that means at least a couple of these biomarkers changed. Like that's how sensitivity is. So it's like, that's where the sensitivity come to the play. But we made sure, because we wanted the clock to really behave, so we did a lot of like the testing and made sure statistically we made sure we are not overfitting, and this can be generalizable. So and that's because we wanted both a specific and sensitive biomarker that we can assess our interventions.
Yeah. And this is actually kind of an important question in some ways, right? So you actually showed that the EnsembleAge outperform previous clocks in both detecting sort of rejuvenation and stress. So what does this really mean in practical terms, for example, for interventional clinical trials or even preclinical studies?
Yeah, I will talk about the preclinical studies for the moment, because the main application of Ensemble for the moment, they are optimized to detect change in mouse. So it's like, I have a human version, but that one is just as a proof of concept. We can talk about it later. I've noticed it's like a lot of experiments that we do, like the collaborators wants to check the changes in epigenetic. We have many clocks. Sometimes actually we get to the danger of like cherry picking based on the patterns that we want to see, or sometimes like nothing changed. So it's like many of the clocks doesn't show any change. So the question is like, can we have a really reliable, so it's like minimize the false negative. So actually the false positive findings change, of course, with cherry picking was so easy. But if none of those clocks respond, it was hard to say like, what's the effects of these interventions on epigenetic. So that's why it's like this comes to the play, because it's sensitive, it can say if this intervention, these specific chemicals, even if it might have a small effect on epigenome, but it can detect it. And because as you will use a biomarker, one of the main application of biomarker is to assess what's the long-term effects. So you don't need to do like a lifespan experiment, like a survival experiment, or like the comprehensive like frailty assessment. With this, you can quickly screen, and then you go to the deep phenotyping. So it can really reduce the animal numbers as well when it comes to the experiment.
Yeah, like you mentioned cross-species translation. You also introduced human mouse, as you just mentioned, and we could talk about it maybe a little bit. So why is this important, and how did you manage to build this, since it's not, the same as having all those perturbations as we have it in mice and all the different organs as well?
Yeah, that's the human mouse version. Unfortunately, I didn't have access to all the human cohort studies. to really carefully benchmark this. But I have access to some human samples, around 80 of them, which we use the mammalian array to generate the data. So the mammalian array, as I mentioned, is measures DNA methylation in the conserved region. So it can be applied to both mouse and human. And around a couple of thousand of those probes, maybe 2,000 to 4,000, so it's like less than 10% of those probes also shared with epigenetic, which is one of the human gold standard array that people use and generated data. So it's really a small portion of the CPG is shared across everything. And when I looked at them in the system, they have high quality in all of them. So that means it's like the mammalian array. It doesn't matter if you run it on mammalian array or epic, you will get the same data. So I said, okay, so we have these shared CPGs. So now there is opportunity to build something that potentially also work in human cohorts. But I don't have high hope. But it's like I just generated as a proof of concept, because in my mind, it's so hard to build something like EnsembleAge in human, right? So because it's like these are experimental data. In human, it's epidemiological data and potentially a few clinical trials, but it won't be at this scale. So it's like maybe the mouse experiments is a better way to benchmark epigenetic clocks and then test the interventions in human, especially if the intervention is relevant. But we have a platform issue here. So it's like, how can we break the platform difference? It's like the data generated in human is a different platform from this. So it's like that's what we have.
That makes sense. One question, I guess, which is really interesting in some ways, is there have been mouse clocks in the past, right? But they have typically focused on sort of predicting chronological age or maybe even frailty. But this would arguably be one of the first mouse clocks that can predict sort of time to death in some ways, right? So what's the implication here? And are we finally in some ways seeing clocks that can really predict mortality prediction in mice?
Yeah, so you see an example, some big data sets we have about the time to death, right? So the easiest actually, if you want really time to death, I don't think clock is the best thing, because it will just say it's like, it, of course it correlates with time to death, but it cannot say how many months or days or years to that. So you can directly build a time to death predictor, and which is much more numerical than the quantitative. So that's one comment. But the thing is, it's like the advantage of EnsembleAge. EnsembleAge is not a predictor of age anymore. It's really predictor of a calibrated age. So we calibrated age based on the health outcomes, right? So it's, in my mind, it's a surrogate of health or like potential longevity rather than age. And you should know, in the training set, the only animals I included in the training set, it's the one that receive the interventions. So it's either a stress animal or like the animals receive a kind of rejuvenation intervention. So there is no healthy control animal in the training set of the ensemble. So that's why it's like it really maximized the sensitivity to respond to different interventions.
That's really interesting. So did you notice like any of the interventions that were better for those animals than others? Like there was some that responded, before I jump into the next question, just out of curiosity.
So actually I learned a lot about the underlying biology of different interventions. So of course it's like we could rank them. There are many of them that work great. Like for example, the calorie restriction. So it works, all of the clocks in the dynamic ensemble are responsive to. But there are interventions that are so specific, so only few clocks respond, or even some of the clocks respond in the opposite way, so the paradoxical. One example, for example, is this progeria mouse model we called Lucky Mice, which is this Hutchinson-Gilford progeria model. So they show progeria phenotype. So of course they age fast, and they show many of the phenotypes that people see with aging. But interestingly, when you go to their epigenome is at a much younger state than the normal age matched mouse. So it's like, so this laminopathy actually caused the epigenome to push them to the younger state, but that's not a healthy younger state of the epigenome. So that's why a lot of clocks really suffer to measure age acceleration in these, because the biology behind it is so different. So that's where it's like when you build a sensitive clock that really track health rather than the highest correlating signatures of the chronological age, then you can find it.
Yeah.
That's it.
I guess, like, just continuing now in the question, you also performed some epigenetic wide association studies on ensemble age acceleration and uncovered a strong signal in promoter regions and chromatin states that were tied to this lifespan regulation. What was the main stood out of this on your results?
So a couple of interesting things. For example, when I looked at the time to death in the mice, interestingly, many of the signatures, as opposed to human, because these are controlled mice in the lab, many of the signatures that relate to time to death actually relate to age as well. So it's like that's an interesting part. So it's like changes in the promoter or heterochromatin is pretty matches age. When we do this cross-ensemble age analysis, also, of course, there is overlap with age, but there is also a lot of unique signatures that are coming up that cannot be found when you only look at age. So these are the insights. So it's like the point I want to make is like the ensemble age approach really brought up new insights on the epigenetics of health in mice that we couldn't get on by only looking at age or even time to death in the mice.
That's really interesting. So one question, I guess, is do you see EnsembleAge as just a measurement tool that can be used to sort of guide therapeutic development, or is it something that can actually be also used to discover targets? Because, of course, you have all of these clocks and that are affecting different DNA methylation regions. Do you see some possibility of it also being used for target discovery, or is it specifically as a measurement tool for now?
No, it's 100%. It's great for discovery, right? So it's like my work is preclinical stage. So it's like this is a screening tool
Yeah.
because it's being used for mouse, right? So that's where the targets comes in. So in the R package I've also released all the plots. So it's like around 50 of them in mouse.
Mm-hmm.
I labeled them based on different interventions. So the reason I release it, because it's like, I think, of course, when it comes, if you want to assess the final outcome of your interventions, like potential, it's like look at the static version. But if you want to see, is it similar, for example, to more like a caloric restriction or like a genotype like growth hormone, you can go deeper into individual clocks. So it's like these are and see it's like what kind of biology your intervention covers. So if it makes sense. So it's like that's how it can guide and say, okay, so for example, my biology is more the intervention I did is more similar to, for example, caloric restriction or the metabolic change. So that's what I'm covering, but the other clocks are not that much sensitive to it. So that means this is, and then of course I'm sure it will lead to new type of combinatory interventions or like.
Yeah.
Yeah, that's really, really interesting. So I guess like the lingering question, I know that you touch a little bit on this and how noisy human data is and, the issue of, the platform disconnecting here. But I guess the whole point is how can we expand this to do in humans so we can have the equivalent of EnsembleAge in humans? So what data sets or, do we have to move into a whole genome screen and drop the arrays or, what collaborations are we going to need to establish to be able to come up for a human version of this?
So I think the human version of this, of course, I introduced this dual clock. I don't know, test. Rawat himself actually worked on human data a lot, so he should be able to test it and tell me, no, it doesn't work at all. So, but it's like just show me how it works. But of course, actually, there are multiple ways. So one way is like completely develop a kind of methyl gauge in human. So that will take time. That means generating data in clinical trials. So it has to be actual clinical trial. So the intervention, it's in the controlled study design. So it's like you really cover it for all the confoundings and then you see potential effects. So it's like that's the best way. So you would know what's the phenotypic effects now, and you have a DNA methylation data. As you build this, then you should be able to come up with a biomarker that is responsive in expected direction in all these benchmarking interventions. So that's one way. Another way, I think, actually, if there is opportunity to generate data with the mammalian array in human, So a couple of cohorts in human that like you have the time to death. So actually the couple of cohorts that have the epic array, like you have a good understanding of the underlying disease and genetic. And maybe it's like if some of the collaborators generate data in the mammalian array, then maybe these mouse data, it's a good way to benchmark the clocks that we think work in human, because it's like this is right now the clock I introduced is only based on I think only 2000 CPGs. But if we had the mammalian array, then the coverage will be around 30,000 CPGs. So it's like we could have a good coverage across the genome, across different genes. And then, of course, it's like it will increase the chance of success in human.
Yeah. I mean, this is outstanding work, I have to say that, and
Mhm.
EnsembleAge sort of feels like a much needed evolution in the clock space. Well, thank you for joining with us today to share this work.
If you want to dig deeper into the EnsembleAge, we'll link the full paper in the show notes. Don't forget to subscribe and share this podcast to your fellow BioAge friends and whoever might be interested in the field of aging itself. So thank you so much. I mean, it's been a real pleasure to have you in the show, and I'll see you next time.
Sam,
Thank you. Thank you very much. It was a pleasure.
thank you, Amin.